Hello, E3 Scholars. Recently we started talking about cell communication, and in our last video broadcast we were looking at just the receptor portion, either the membrane proteins, G proteins, tyrosine kinases, gated channels, or the intracellular for the steroids and the small hydrophobic molecules that can go through. The second step of that pathway, as we looked at quickly, was signal transduction. That's the pathway that can help amplify, control, or regulate the signal, help fine-tune it more, especially in complex. But this transduction is beneficial for both single and multicellular animals or organisms. So it is a benefit. Again, the greatest benefit is amplifying the signal so that you don't need a large quantity of chemicals to make it work, or the ability to coordinate and regulate and fine-tune the process. Anyhow, think of dominoes. That's really what it is, and we'll get to a picture of it, but it's like dominoes, that you just have these proteins, and you are in most cases phosphorylating and dephosphorylating to turn on and off that switch to get the signal activated and amplified. As we will see with responses, you can use epinephrine, and by the way it works, you just multiply that 10 to the 8th times from what you start with, so you don't need a huge signaling concentration of epinephrine signaling molecule. All right. So, and here's what we're looking at. Here comes the signal molecule. It binds the receptor. This is a membrane receptor. You've activated the relay molecule. That triggers the protein kinase, okay, which phosphorylates that protein, which triggers the next, So you can see you're getting phosphorylated in the process. After it's phosphorylated, the next one, it gets dephosphorylated and goes back to inactive, so it's ready to use again. Eventually, in this pathway, you'll activate a protein that creates the cellular response. And we talked about kinases before. They are enzymes that work with proteins and stuff. The phosphatases remove the phosphate so it can be reused, but that 2% of our genes may code for kinases just because of these pathways that help amplify and fine-tune the signal. Now that's a direct pathway. Not every pathway or not every messenger is a protein. Remember, proteins are large and they're difficult to diffuse. We saw with G proteins that you can have diffusion in a small area, but non-protein, water-soluble molecules, ions and stuff can diffuse much more rapidly through the cell, and in that transduction, and really speed up the time of that. And with the phosphorylation and the cascading thing, as we've seen before with cell respiration and photosynthesis and stuff, can really amplify the signal. The most common one is calcium, but cyclic AMP is also those are the two biggest. They work with both tyrosine kinase and G protein receptor molecules. CAMP is one of the most widely used. Adenylyl cyclase is the enzyme that converts ATP to CAMP. There you go. And then phosphodiesterase, with a little hydration synthesis there, creates it back to AMP, which can then go back to ATP. Okay. CAMP is big. Epinephrine was used. They saw with epinephrine in liver cells that CAMP concentrations went way up. What they usually do is activate something called kinase A, protein kinase A, which phosphorylates various proteins. Okay, and then so you can see here. Here we go. Here comes in your first messenger. These are called second messengers because the signaling molecule is always the first messenger. And then anything after that is called the second messenger. So G protein, GTP activated, hits the enzyme. This is the adenylyl cyclase, converts ATP to CAMP. That activates the protein kinase, which then goes to the cellular response. Of course, this enzyme would also then be regulated to deactivate the whole system also. Okay. So that's the second messenger. It's a much faster way of doing things. It's really using the same receptor, but in the transduction pathway, it allows that to crank up and be much faster and amplify the signal. Calcium is really the largest of all. It's used throughout the body, and it also involves using what's called an inositol triphosphate and diacylglycerol. Let's show you here. Here's what we have. Because calcium, the concentrations of calcium intracellularly are very, very small. Part of that is the diffusionary need of the endoplasmic reticulum. Part of that is protection. If this were a muscle cell contracting, the calcium with inorganic phosphates around would create what's called hydroxyapatite, which could cause muscle cells to start calcifying. So the concentrations are kept very small, 10,000 times less than what's flowing around in your bloodstream. Anyhow, here's our first messenger signaling molecule. It's a G protein. Again, activated the G protein with GTP. This in turn activated what's called phospholipase C. This is an enzyme that goes. This PIP2 is just a membrane phospholipid. It cleaves it here. And what you get is the formation then of the diacylglycerol and what's called inositol phosphate, the second messenger. Now, that then activates the calcium channel ligand gate in the endoplasmic reticulum that releases the calcium, which diffuses through. This is the second messenger that activates various proteins, and you get a big cellular response. And you say, well, wait a minute, this was the second messenger, this was the third messenger. The first messenger is always the signaling protein. Anything else that works in there is a second messenger. They don't distinguish to 3, 4, and everything else. But what we will see here in our cellular response then is that these things can flow much faster through the cytoplasm and trigger these actions than can a protein. And again, gives it amplification and fine-tuning methods, because if you think of almost no calcium here and a lot of calcium here and the diffusionary thing, then you can activate a whole lot of proteins at once. Anyhow, that's the idea of what a transduction pathway does, either through second messengers or phosphorylation, is take a very small signal, amplify it, speed it up, and then get singular or multiple cellular responses. Our next film in the series will be What is that response? Until then, Scholar on Eastview.